All right, cell membranes. I think that most of you really like cell membranes because we made those models and everybody seemed to understand it really well, which was awesome. As a reminder, cell membranes are a fluid mosaic model, meaning it's not like stiff. The cell membrane is constantly moving and flowing. It's a fluid. Let's talk about all the components of the cell membrane. There's phospholipids. I showed them in pink. There are saturated, which is straight, and unsaturated phospholipids, and this helps with the fluidity of the membrane depending on the temperature that it is. The majority of the cell membrane is made of phospholipids. As a reminder, phospholipids have a polar head group, meaning attracted to water, can have a charge, or does have a charge and can interact with charges, and the hydrophobic tails, meaning they're afraid of water or they're nonpolar, they don't have a charge. Proteins, there's integral proteins which go all the way through the membrane. This can include channels or pumps or just structure proteins, and peripheral proteins, which would be more just on the side of the membrane. For proteins that are integral proteins, they would have a polar area or two polar areas, hydrophilic areas and hydrophobic areas. So the R groups on this part of the protein would be hydrophobic, which would allow them to be with the hydrophobic tails of the phospholipid. Okay, also there are glycogroups, like on glycoproteins or glycolipids. I showed a glycoprotein here where we have a carbohydrate group attached to a protein. There are also glycolipids where a carbohydrate group would be attached to a lipid. These can be used in cell signaling and sometimes in anchoring of cells to a certain location. There's also cholesterol. Cholesterol is a type of lipid. It's usually you'll see like 4 circles if cholesterol is being represented. And cholesterol helps with fluidity, meaning it helps to make sure that the cell is the right amount of flowy depending on the temperature. All right, membrane transport. So cell membranes are selectively permeable, meaning certain things can get in, certain things can't get in. Small nonpolar molecules like carbon dioxide can pass straight through the phospholipid bilayer. Also, oxygen is another example of a small nonpolar molecule. Larger and polar molecules typically need a channel to go through. So something like glucose or maybe a signaling molecule. And water, some water can get through the phospholipid bilayer, but the majority of water needs to go through something called an aquaporin, which is like a pore for water. As a reminder, passive transport is when we're going from a high concentration to a low concentration. Naturally, molecules are going to find their equilibrium, so they're going to slowly diffuse. What's the word? I've forgotten the word. Where the molecules are actually going back and forth, but over time we end up having an equal distribution. So we call it, I can't remember what we call it, but it's always moving. The molecules are always moving, but we end up with an equal distribution. Active transport is when we're going from a low concentration to a high concentration. We need to put energy in to make that happen. ATP is the form of energy that is used. So if ever there's a low to a high concentration, you know we need ATP. And this is called against its gradient. Passive is called with its gradient. 2 examples of active transport that don't look quite like this picture are endocytosis and exocytosis. And those words tell us what they do. Endocytosis means in. Exo means out. Endocytosis is when the cell takes something in. So, for example, maybe this is a piece of food, and that food goes to the side of the cell. The cell decides to engulf it and take it in, and it's then in a vesicle. That vesicle might merge with a lysosome that has hydrolytic enzymes that would break that food down. That's one example of endocytosis. Something like phagocytosis is an example of endocytosis. Exocytosis is the opposite direction. So maybe a protein was just made. It's coming from the Golgi. It's put in a vesicle, and that protein is sent to the membrane to be excreted. Maybe this is a signaling molecule that needs to go to another cell. And both of these, as I said, require ATP.